presentation of two projects. One is the Seagrass project, that is an Italy-Israel project. The other one is the Scenarios project, that is a German-Israel project. And there will be 4 talks, 2 related to Seagrass and 2 to Scenarios, although the 2 projects are really completely interconnected. So as you will see, we took the opportunity of having these 2 projects founded by Israel, Germany, and Italy to develop a common interconnected scientific strategy that will be presented in the next 4 presentations. Okay, the Seagrass project is coordinated by myself and Gideon Winters in Israel, and the Scenarios project by Mirta Tichberg and Auke Rüther in Germany, and Gideon Winters in Israel. Gideon is the link between the 2 projects. We can start with the first presentation that will be given by Alex Santillan, my collaborator in the Seagrass project. And Alex will present you the general overview on the project and the results we obtained with our experimentation. The title of his presentation is Searching Early Warning Indicators in the Mediterranean Seagrass Posizione Oceanica. We have about 18-20 minutes for each talk. So I will switch on my camera when there will be 3 minutes left. After 15 minutes, I will switch on my camera. So Alex, it's your turn. You can share your screen and you can start. Since then, I will take 15 minutes time and switch on my camera. Alex?
What happens? Maybe microphone. Maybe the microphone must be off.
Hello, can you listen to me?
Okay, Alex. Yeah, you are there. Okay. You can start.
Is the screen Can you see the presentation?
Yes, we can see everything. You can start.
Yeah, I don't know. I can't switch the video on, sir. I'm trying to, but okay. Okay, good afternoon, everyone. My name is Alex Santillan. I am a postdoctoral researcher at Stazione Zoologica Anton Dohrn, and today I'm going to present the work that we have done as part of the project Seagrass, application of molecular tools for detecting early signals of stress in Israeli and Italian seagrass species. This project was possible thanks to the collaboration between Israeli and Italian lead scientists Gideon Winters from the Dead Sea Araba Center and Gabriele Procaccini from Stazione Zoologica Anton Dohrn at Naples, Italy. In this presentation, I will first give an overview of what seagrass are and their importance to the marine environment. Then I will introduce our organism of study, Posidonia oceanica. Then I will explain the interactions between the different parties involved in the project. Later on, I will explain our experimentation to achieve the proposed goals of the project. And finally, I will present some findings from our experience. So what are seagrasses? Seagrasses are a group of superior plants that have recolonized the sea from the terrestrial environment. They are present along the coast of all continents except for Antarctica, and they have a typical morphology of a terrestrial plant, that is root, stem, and leaf. The roots and stem are usually buried under the sediment, and the leaves have a typical ribbon-like form or shape of a grass that constitutes the meadow. This project has studied the 2 species at the extreme of this diagram, and which is Posidonia oceanica, which is a common Mediterranean or the main species in Italy, and this is the biggest in shape of all the seagrasses, and the other one is Alophila stipulacea, which is the most representative in the Red Sea in Israel. and is a tropical species and is the smallest of all the seagrasses. So seagrasses provide a range of ecological roles, such as exporting organic material. They also protect the coastlines by raising the sediment level. They host a great diversity of marine animals, from grazers to top predators, being also important nurseries for fisheries. And they are gaining more attention recently due to its importance and their capacity to store carbon. The economical value of ecosystem services has been estimated in around $19,000 per hectare per year. But unfortunately, these precious organisms are in decline worldwide due to the increasing pressures of human activities. So seagrasses or other marine biota are threatened by the ongoing changing atmospheric conditions. The rise of CO2 concentrations will cause an increase in temperature and the acidification of the seas. On the other hand, more direct human pressures, such as the runoff of nutrients from agriculture and from human waste, are also threatening the meadows that are growing really close to the coast. And also they interfere directly with the cellular process. Posidonia oceanica is an iconic Mediterranean seagrass. Their meadows are highly productive and are the habitat of many species of fish and invertebrates. Some meadows are thought to be among the oldest living organisms on Earth. But unfortunately, as with other seagrass species, they are disappearing at accelerated rates due to the interaction of many direct and indirect stressors. To explain the goals of the project, I first would like to show this conceptual scheme of how ecosystem and species respond to stressful environmental drivers. All ecosystems have a range of resistance beyond which they may suffer irreparable damage. Our duty is to find an early indicator before they pass this non-return point. And on this side, in this diagram, we can see that the different indicators has a different ability to predict this mortality of the seagrasses, of the species. And we can see that the molecular indicators are the ones that are fastest in the response before we see a high mortality of the species. So the main goals of this project were the identification of key genes and the molecular physiology mechanism associated with the heat stress and nutrient excess, both independently and in combination, and the identification and validation of early warning indicators of stress to be used as molecular toolbox in monitoring and management of Italian and Israeli seagrass meadows, with a potential application worldwide. To achieve these goals, the plants were collected in disturbed and undisturbed meadows and acclimated to occurring conditions in our mesocons facilities in Italy and in Israel in the Dead Sea Araba Center, and they were exposed to different concentrations or levels of nutrients and also different levels of temperature. The level of temperature were chosen accordingly to the natural levels and the regions where they were collected, and also taking into account the future scenarios of climatic and environmental changes. I would like also to introduce the collaborations with our German partner from the Leibniz Center for Marine Tropical Research, that they were in charge of the field experimental part and the modeling part, while we in Israel and Italy were in charge of the microcosmic and mesocosmic experiments. In Italy, in charge of Posidonia oceanica, and in Israel, in charge of Allophila stipulata. Project in Germany was led by Mirta Teichberg and Dr. Hauke Reuter. And this created a synergy that helped us to put different expertise in order to improve the prediction of seagrass reaction to environmental change. We chose two middles for Posidonia oceanica for our experiments. One middle was a pristine, considered a pristine environment with a low impact from human activity off the coast of Ischia, the Ischia island. And also we chose a middle in an impacted area close to the town of Bacoli. These both sites are in this area, so they are subject to a similar pattern of temperature. So after the collection at the sea of the plants, they were immediately transported in coolers to keep them safe, and they were prepared in baskets before they being placed in the glass aquaria. The glass aquaria are designed to keep the plants with the right amount of light and also to provide the water recirculation so that we mimic the conditions at the sea. Our experiment consisted of 12 aquaria. In each aquaria, a basket with plants from the pristine side and a basket with plants from the impacted side were placed, and the aquaria were divided in 4 conditions. A control condition where the temperature was kept at 24 degrees, a heat conditions where temperature was raised to 30 degrees Celsius, a nutrient condition where a fertilizer was added weekly to increase the concentration of nitrogen and phosphorus, in a condition where these two stressors were combined. As you can see from our experimental schedule, we gave first an acclimation period so the plants could get used to the new environment. Then we slowly increased the temperature to reach the 24 degrees, that was our control temperature. And also, in a similar way, we increased slowly the concentration of nutrients, and then we added, after each week, an amount of nutrients to keep the concentration of nutrients high. We also have sampling at different time points, that they were after 1, 2, 3, and 5 weeks of exposure to these conditions. At each time point, different parts of the plant were preserved for successive analysis. For example, we extracted DNA, RNA, the pigments. We also studied the carbohydrates and other parameters that will give us an indication of stress. For example, for the gene expression analysis, we collected the shoot apical meristem at this part and froze it at -80, and we also collected a portion of the leaf to extract the RNA. This is actually how the sampling looks like, and this is all the team of Dr. Procaccini working simultaneously to quickly collect and clean and freeze the samples in order to maintain their integrity. And from the first results of these experiments that we performed in Posidonia oceanica, we have recently published in the Journal of Frontiers in Marine Science. So I take this occasion to invite to whoever is interested in the details of this experiment to read our paper and give us your feedback. I will just present a couple of results, the main results from this first publication that are the most representative. The first is the photosynthetic performance that was assessed by measuring the chlorophyll fluorescence and the leaves. And as you can appreciate, there were differences between the populations in the photosynthetic performance, and also among the different treatments within each population. The population of Ischia had the lower photosynthetic performance after 5 weeks of exposure to the treatments, and particularly under the nutrient treatment. We suggest that this is caused by the a diminution in the light amount that arrived to the plants due to the excessive growth of epiphytic algae because of the increase of nutrients in the water. Another interesting result is the different distribution between the soluble sugars between the plants from pristine and polluted sites. So we can see that there was a different distribution of the sugar amounts between the two parts of the plant, the leaf and the rhizome, and that was different in Bacoli, in the population coming from Bacoli, and the population coming from Ischia. Apparently, there's a strategy, an activated mechanism allowing the rapid redistribution of the sugars reserves in the plants from Bacoli in order to cope with the nutrient excess, because the sugars are needed to assimilate the nitrogen. A second phase of our project included the identification of early warning molecular signal of stress. And to this end, we have performed an analysis of gene expression of the whole set of genes of Posidonia oceanica, also called the transcriptome analysis, at the time point 2, which will be an early signal, and then we correlated to the samples and the parameters of time 4, which were the ones presented in the first publication. We used a technology called the tag sequencing using the ion torrent technology. So we analyzed two different tissues, the meristem, as I explained before, and the leaves, and we could see that there is a differential response in the response to each plant tissue, and also from different populations, Ischia and Bacoli. Using all this data set of the gene expressed, we have performed a gene co-expression analysis, which groups all the genes with a similar expression pattern into modules of co-expression, and these modules of co-expression will indicate us which genes are the more involved in the response. So after performing the identification of the modules, which are assigned a different color, we correlated with the responses observed at the time 5, at the sampling time 4, after 5 weeks. And then we have a correlation index and a statistical signification, and we're gonna select these modules to pick up genes inside these that are important, for example, for the survival of the plant. The selected genes have now to be tested and used in analyzing the gene expression from the field samples performed.
3 minutes to go, Alex.
Okay, for the field experience of our colleagues from our German partners that performed the field experiments, and using the technique of the qPCR, which now is famous and popularized due to its use in the diagnosis of the COVID-19. And I will highlight, I will take this opportunity to highlight that the pandemics has caused serious delays in the development of the project and the laboratory work. And then we fear that we will not be able to test the selected genes in the field samples before the deadline of January. So we will probably ask an extension to be able to successfully achieve and test and complete the goals of our project and validate these genes that we have found with the analysis of gene expression. I would like to thank the funding from the Ministry of Foreign Affairs of Italy, the Ministry of Science and Technology of Israel, and the support of Stazione Zoologica Anton Dohrn in the sampling campaigns. And I would like to invite you to visit our website if you are interested in the research we do. Thank you very much.
Alex, thanks a lot for being perfectly in time. I didn't say at the beginning that we save the questions at the end of the presentation of each different project. So I would pass to the presentation, to the talk by Gideon Winters from the Dead Sea Area Science Center. And Gideon will tell us about predicting the interactive effects of heat waves and eutrophication on populations of Olophila stipulacea from the northern Gulf of Aqaba, a system biology approach. Gideon, again, after 15 minutes, I will switch on my camera and you will have 3-4 minutes to complete your talk. So you can start. We can see your screen.
Okay, thank you, Gabriela, for the introduction. Can you hear me well? I hope. So this project was funded by the Israeli Ministry of Science and Technology, and it's a joint effort from our institute, which is in the middle of the desert, not very near to the sea, in the Dead Sea Arava Science Center, and Professor Simon Barak from Sde Boker, Ben-Gurion University. So let me just start at the beginning, because it's a bit more easy in Italy to explain to people about seagrasses, but in Israel it's more difficult because within the Gulf of Aqaba, it's actually coral reefs, which are the major focus of scientific studies, academic courses, monitoring programs, and major efforts for conservation and restoration. Seagrasses get very little attention in the Gulf of Aqaba, and this is quite surprising because they have very important ecosystem services, which Alex spoke about, and some of these system services are very important for corals that are living nearby. So it's surprising that so little attention has been given to seagrasses. The most dominant seagrass in the Gulf of Aqaba is this species. This is a small, tiny species, tiny compared to Posidonia. Posidonia can be more than 1 meter. This is around 5 centimeters. It's a really tiny seagrass. Sometimes it's the dominant species, but sometimes it's the only species. And we made a map a few years ago, and it's found all along the coast here in green, nearly throughout the whole coast. Here's the border with Aqaba, with Jordan, and here's the border with Egypt. So it's found throughout the whole coast in the Gulf of Aqaba. It covers a large area, and it's worth around $2 million per year in ecosystem services. And then that's how you get the attention of people in charge of conservation in the region. Just to show you how it looks like, a quick movie. Again, this is a very small plant, very different from what Alex showed you before. But again, this is another movie. I hope you're looking at the seagrass and not at the stingray. But this is how the habitat looks. This is in the North Beach. A very extensive meadow, small plants, but very extensive meadow. But the seagrasses in Eilat are being threatened at the same time by global and local stresses. This is a paper we published this year showing data for 35 years of climate change trend in summer and in winter. And what's really surprising is the annual trend is faster than the worldwide global average for coastal regions. So we are experiencing climate change, which is happening faster than most places around the world. That's a global threat, of course. But we also have, at the same time, local stresses. Eilat, as you maybe know, is a major tourist attraction site. And because the physical structure, the physical structure of the Gulf, which is basically a semi-enclosed basin, there's a long residence time of its local waters, 3 to 8 years, which means that any pollution event is stuck in the water for many, many years before it disappears. And not only this is a problem, but because it's a tourist site, there's major development, coastal development projects in the region, which are expected to increase the nutrient and the organic loads into the marine environment. And eutrophication, does it make thermal stress worse, or does it react the same for all populations? Are all populations equal in their response to thermal and eutrophication stress? We don't really know, because most studies on seagrasses, on stress in seagrasses, focus on one population exposed to usually one stress. And this doesn't really mimic the situation in nature, where we have a realistic scenario where plants are exposed usually to more than one stress at the same time, combination of stresses. A point on seagrass monitoring, we are very much interested in contributing early warning indicators, as Alex explained before, because most of the monitoring of seagrasses is done by following changes in community structure. You look at percent of cover, changes in biomass and species composition. The problem is that by the time you've found those changes, this is usually too long. It's a very long time to respond until you see stress has gone through the system and taken many, many months and sometimes years to change these parameters. So there's a growing interest to develop early warning mechanism for stress. So that's the main motivation in that part of the work. And this is our model where we have plant response on the y-axis here and a status of a population here, declining population. Most of the parameters that are involved in monitoring are around here. We have a bit of morphological responses, which are a bit earlier, a bit of physiological responses like PAM that Alex showed, which is a bit earlier, but can molecular responses give us a much earlier indication of stress? So that's where we're heading to. And maybe if we can find these, we can help the system a little bit. So our main objectives in this C-stress project were to understand the thermal tolerance and resilience of populations and to temperature with and without nutrients, and to develop early warning indicators. So where did we do this? We went to Eilat. We took 2 populations, sampled plants from 2 populations we have earlier projects on, and we've shown earlier that they differ quite largely in their amount of light which they receive here, and there are different sediments in these 2 sites, and because there's different amounts of light, the plants actually react differently. Even the same depth, they have a much larger surface area in the more polluted site, Turiam, compared to the more pristine site here on the South Beach, which is a more cleaner site, less nutrients, and of course, so the plants here have smaller leaves. Although we are in the middle of nowhere in the desert, we built a marine ocean in the desert where we have 20 aquariums, 20 tanks. We have 4 systems where we have one which stays at 27 degrees, one which stays at 27 degrees but is nutrified, so we add nutrients in this system. We have one which simulates climate change and is exposed to 31 degrees, which is around 5 degrees warmer than their natural conditions. And we have one which is exposed to both temperature and pollutants. And we have 2 populations in the same aquarium, just like Alex showed before. So this is our profile of experiment. The plants came in here. This is the temperature treatments. They went slowly up to 31 degrees. Control stayed at 26.7 degrees here. And then the heat stress came down. So at the same time as we're exposing them to thermal stress here, we're also adding from T0 onwards nutrients into the system. And we made all these measurements. I won't show them all, but we'll just talk about 2 or 3. So the first measurement that I'll show is how much did the plants grow. This was done by adding a plastic cable tie and actually following this over time. So what we see here is the same temperature time profile I showed before, but here we see the treatments. 27 is blue, 27 plus nutrients is this dashed color, warm is red, and warm with nutrients is dashed red. What we can see here is that already in T3, the plants with a warm temperature actually grow faster. But it seems that the nutrients are actually stressing the plants from the South Beach, and it's causing them to slow down their growth. You can see this following here, and following here. Recovery seems to have a full recovery at the end here. This is quite different in the Turiam, which is the more polluted site, where they don't seem to care so much about nutrients, but they do care about nutrients and temperature much more. And there's less recovery at the end of the experiment of the plants from the polluted site. So these are the 2 interesting time points. But we find the first stress at T3, so that's around 2 weeks or 3 weeks of thermal stress. We went to PAM fluorometry, so this is an indication of how good the photosynthesis is doing in the plant. And the first sign of some sort of stress is found in T3, where warming actually increases the quantum yield for the beginning, but nutrients in the clean site affect the reduction in the quantum yield. So the plants in the clean site are not so happy from the nutrients. And you can see this even more here. With nutrients, thermal stress is not such a big deal, but thermal stress with nutrients or nutrients by themselves is a very big reduction. But again, a nice recovery at the end of the 1 month of recovery stage. This is quite different for the Turiam, where they don't care about the thermal stress here in the red, but give them nutrients, be that with cold water or warm water, and there's a vast reduction in quantum yield. And again, not much of a recovery here at the end here. So if we look at mortality now, we see that the first sign of mortality in the South Beach populations is around T4, so around 1 month or 3 to 4 weeks of thermal stress. And again, you see the dashed line, so they're dying more from nutrients than from temperature. You see this again here, dashed lines, and here dashed lines. But their Turian population, which is a polluted site, is actually affected much more by temperature than by nutrients. So these red ones here and the red dash plants here. So to summarize this part, what are the implications of these results so far? It seems that the population's nutrient history is important in predicting its resilience to thermal stress. Pristine nutrient-free populations might have a better chance to recover from other stresses, for example, temperature. And we think we've shown you that eutrophication enhances its synergistic effect, enhances the effects of already a thermal stress. So this has implications for how do we develop water quality levels based on these results. Some molecular responses, what we see here are some Venn diagrams, and I'm just going to point I won't go into them too much into details, just point the fact that these are genes here which are variability between tanks. So we have a bunch of genes which are responding to the variability between different tanks, different plants. Because the plants are aging, we're getting genes which are expressed differently in the same treatment, but over time. So these plants are getting older and they're expressing different genes. But if we look at these ones, these are genes which are, because of the treatment, 31 degrees compared to 27 degrees. And some of these genes will be early warning treatment genes. If we compare Turian with South Beach, we find that the South Beach populations are expressing around twice or more amount of genes, 3,000 genes compared to 1,700 genes due to thermal stress. So they're more responsive to thermal stress than the polluted site. And without going into too details, we see the same picture in the nutrient analysis. When we look at the transcriptome analysis, we see again more than nearly 3 times the amount of genes responding in the South Beach, the pristine site, to the nutrient stress compared to the polluted site. So we see a big difference here between here and here. This is a major complicated diagram, but this is a GO-term biological function enriched GO-term analysis. And what we found here is that there's 32 processes, biological processes, involved under temperature stress. Some of them are found in other stresses as well, the nutrient stress. We find 25 biological responses, GO-terms, under nutrient stress. Again, some of them are found in temperature stress. But we find 13 GO-terms which are common across all populations, but they're found only in nutrient stress, and temperature stress. So we have a pretty good eye on what's actually happening in temperature stress. And similarly, we have around 3 which are going here, 16 GO-terms, which are responsive only in nutrient stress, but not in temperature stress. So to finish this, we found 2 processes which are found in all stresses, which is actually quite low for plants. These are they're found only in double stress. This is a core stress processes, maybe like oxidative stress or things like this. But basically, this is a pretty low number of functions which are shared or common across all populations and all stresses. This is one example in my last slide, I think, on what's an early warning indications gene, an example. Let's call this gene 161, for example, here. We see that in T0, there's no difference between the treatments, of course. There's no difference in the expression. A week into the first day of thermal stress, T1, we're getting a little bit difference between the control and the treatment. But 1 week into the treatment, we find a huge difference between the 31 degrees and the control 27 degrees. And as we go into T5, we see, again, a big difference between T5 expression and T5 control, and it goes down in the recovery. So this would be a good candidate for early warning T2 1 week into the stress before we see any other signs of stress in the experiment. So to summarize, again, like Alex, we've had many backlogs because of the pandemic, but the Venn diagrams show you that seagrasses from the pristine sites are more responsive to both nutrient and temperature stress than plants from the more polluted site. We have identified genes which are only affected by one stress, and genes which are affected by double stresses. But now we're cleaning the data and we're looking for early warning stresses that are either up or down regulated, and they will be used as a finally early warning stress indicators. And this is our big team. I'd like to thank all our members, and thank you very much for your time.
Gideon, thanks. Thank you very much, and very, very interesting results. But it looks like we are having similar results in response to nutrients and temperature, like nutrients are affecting more. And the nutrient history of populations has a huge importance in the capability of population to respond to stress. So we are together on the right track. Okay, we have time now for questions for these first two presentations regarding the Sea-Stress project, and also people connected in YouTube can ask questions, and the questions will be directed to us. So please don't be shy. Anything you want to ask, and any curiosity or maybe idea and anything, we are here for answering to your questions. Alex and Gideon will be happy to answer.
Alex would be.
Otherwise, maybe we can, if there are no questions at the moment, we can pass to the second set of presentations, and then maybe there will be more questions at the end of the session. And the second project we are going to present is the Scenarios project. And as I told you at the beginning, this is an interconnected project with the Sea-Stress. And the German partner from the Leibniz Center for Tropical Marine Research is going to present two aspects of the same story of the project. And the first to present will be, the first presentation will be by Mirta Tichberg and Stephanie Elber. And I think Stephanie is going to present. And the title of the presentation is Seagrass Reaction to Elevated Nutrient Levels in the Mediterranean and the Gulf of Eilat. So again, Stephanie, you can share your screen, and then it will show up after 15 minutes. So keep the time.
Should like.
Yeah.
Okay. Yeah, so I will present to you today.
I don't see the screen anymore now.
Is it not there? Okay.
I don't know if it's just me, but do the other people see the screen?
I make it again.
Try again.
No.
Okay.
So, no?
It was there for a moment. Okay, wait. Okay, here we are.
Okay, good. So I will present to you today the Scenarios project, which is a collaboration between the Leibniz Center, the Stazione Zoologica in Naples, the Dead Sea and Avara Science Center, and the IUI in Eilat. And Scenarios stands for Seagrass Scenarios under Thermal and Nutrient Stress, and was funded by the BMBF in Germany. So the overall aim of this project was to identify early warning indicators for stress in different seagrass species. And therefore, we conducted field and mesocosm experiments. And you heard Alex and Gideon's presentation, so they were the responsible person for the mesocosm experiments. I was the responsible person for the field experiments, and knowledge obtained from our results should be used for the development of an ecological simulation model. And this model will be developed to give sound guidelines for managers so that appropriate measures can be taken before irreparable damages can occur to the whole meadow. And the fieldwork consisted of two parts. The part one was conducted in Israel with Halophila stipulacea, and the second part in Italy with Posidonia oceanica. And basically, we wanted to simulate in situ nutrient stress. And at both of our sites in Italy and Israel, we chose 2 sites in a heavily anthropogenically impacted area and 2 sites in a low impacted area, or basically a marine protected area. So we established in the seagrass meadows 6 plots with a diameter of 2 meters. And to 3 of them, we added nutrients, and it was slow release fertilizer filled in PVC tubes that were hammered like 50 centimeters in the ground and were also 50 centimeters above the ground to enrich both like the sediment and pore water and also the water column. And to monitor the in situ temperature stress, we wanted to take advantage of the natural heat peak and sample 1 time before and 1 time after. So in Naples, actually, we started sampling in June. You can see it, it's the dashed line, and then again in September. In Eilat, we went for the first time in July, and then again in December. And first, I tell you about the results and what we did in Israel with Halophila stipulacea. So our study sites were in the Gulf of Aqaba. And as Gideon told you before, we had like 2 sites. So the South Beach site, which is a marine protected area, and you can see there's basically nothing. It's really close to the Egyptian border. And our second site was the North Beach area, which is densely populated, or there's like this whole hotel strip, and it's exposed to municipal and agricultural discharge. There are a lot of boating activities. Site 3 is directly next to the marina. And to show you how the meadows look like, the other 2 pictures show the South Beach area. So you can see water is very clear. You have high visibilities. Seagrass is kind of small. And the lower panel shows the North Beach areas. So there you can see it's a bit turbid. The seagrass grows more densely and has thicker, longer leaves. And basically what we also did, we monitored the temperature over the whole time of the experiment, and we logged it every hour. And what you can see now is basically the average daily temperature over the whole time. And basically the highest temperature, as already in former studies confirmed, was in August. And I made the dashed line at 29 degrees because it was shown that Halophila stipulacea populations from the Western Indian Ocean and also from the North Beach started to experience first sign of temperature stress at this degree. And what also is important to say, it was not the surface temperature, but at 10-meter water depth, directly above the seagrasses. And here you can see, for example, the changes in meadow cover. So on the left panel, it's always the South Beach sites. On the right panel, the North Beach sites. And then before the heat peak is what the left window shows. And then it gets split up into control and fertilized plots. And filled circles and empty circles are always the 2 sites in the South Beach, and the triangles, the 2 sites in the North Beach. So you can see that from July to December, there was like in the North Beach an increase in meadow cover, and in the South Beach, it was more pronounced with fertilization, the increase. And when you look at the shoot density, you can see that shoot density actually decreased, and it was less pronounced in fertilized plots, this decrease. We also checked the above ground biomass, so basically the weight of the leaves, and the below ground biomass. There we can see that the above ground biomass and the below ground biomass in the South Beach areas was decreasing, while biomass in the North Beach increased. And with fertilization, there was even a further increase in biomass in the North Beach area, and in the South Beach, a further decrease in biomass. And probably the increase in the above ground biomass at North Beach is caused by the changes in the leaf morphology. So we measured all kinds of morphological parameters, and here you can see the picture that to show you how different the leaves look like. So in the upper panel, there are the South Beach meadows, so they have very small and tiny leaves, and the South Beach and the North Beach meadows have compared to them really wide and long leaves. So we looked, for example, at the percentage of apical shoots in the population. So if you look at this picture, this is where the seagrass starts its growth, and this is like the apical shoot in the red circle. And we could see that apical shoots did not make up so much shoots of the whole shoot population in the North Beach. And basically they stayed the same in fertilized areas, but otherwise increased in control areas. And South Beach had the whole time around 20 to 30% apical shoots in its population. Then what was also striking was we found reproductive shoots only in July, but quite a lot at the South Beach Site 1. And here, to show you how it looks like, here are two pictures. So this is like a seed capsule, which develops after the female flower is fertilized by the male pollen. And yeah, so both the percentage of apical and reproductive shoots was shown to increase as a stress response or an unfavorable environment. And there you can see fertilization. There was a trend of further increasing apical shoots in South Beach, while the opposite was true for the North Beach populations. So North Beach populations actually don't seem really stressed, but the South Beach do. We also looked at the Inter-Nodal Distance. So basically from one node to the other, which shows the red stripe. And this is like also shown that small internodal distances are a common response to environmental stress, and that it's a proxy of increased shoot and or root densities. And you can see South Beach plants also have smaller internodal distances than the North Beach ones. We also checked leaf area index, and you can see it was almost all the time double as high in North Beach than in the South Beach area, but increased with fertilization in both areas. Then we looked at carbohydrates in the rhizomes, so at starch and sugar. And actually the main form of those non-structured carbohydrates was sucrose in the rhizomes, if you look at the axis. And it actually, after the summer heat peak, there was higher rhizome starch content in the South Beach blends, and the rhizome starch in North Beach blends decreased significantly by around 40%. And all fertilized plots showed a decrease in their sugar contents in the rhizome tissues. So in conclusion, like Gideon already mentioned, in the mesocosms, we found the same, that it's really important, the nutrient history for the responses to thermal and nutrient stress of both populations. We actually found that negative effects, especially for nutrients, were found in the South Beach meadows, and the seagrasses at the North Beach seemed to have even benefited from the additional nutrients, probably because they have already adapted to the higher nutrient concentrations and the high water turbidity at the sites. The only concern we found for North Beach meadow was the depletion of the rhizome starch contents, which was by reduction of up to 40%, which is probably the result also of the constrained light penetration during summer months or the beginning of temperature stress. So probably we also looked at isotopic composition in leaf tissues and found that there was a significant decrease in 13C in leaf tissues, which is also identified as an indicator that decreases in response to low light. And also, I mean, for North Beach meadows, until now, probably their rhizome starch is not that important, and they use the energy for their fast growth and invest a lot in their above ground biomass. And so we could show that Halophilus stipulacea is one of the most tolerant and plastic seagrass species, but they might be more susceptible to stressors in its native range, and monitoring efforts need to be increased in Eilat. And for our second fieldwork part, we went to Italy and looked at Posidonia oceanica. So our study site were close to Naples and were actually the same sites Alex introduced. So we had 2 sites in the Gulf of Pozzuoli, which was our impacted area. So you can see this is how the meadow looked. There were a lot of algae growing inside. It was more like discontinuous meadow patches than like a whole meadow. You can see the harbor of Baia, and here the mussel aquaculture farms that were really close to the meadows. Our 2 low impacted sites were sites around Ischia. So at Ischia, you can see it's like long continuous meadows. Visibility was amazing. We could see the divers working at 10 meters from the boat. And it has also like this mat of dead and old rhizome and roots. And yeah, again, we monitor the temperatures at 10-meter water depth. And the dashed line is put at 27 degrees because it was shown that increased shoot mortalities and negative impacts on the photosynthetic rates started to occur at temperatures starting from 27 degrees or beyond. And the highest month again was August. So we had these high temperatures in the beginning and in the end of August. And the maximum temperatures recorded were around 28 degrees. And here the meadow cover, you can clearly see Ischia had at all times a higher percentage cover, and decline in meadow cover was more pronounced in the fertilized plots, so in the yellow dots and triangles you can see here. What was most striking was the response of the epiphytes. There in plants of Ischia, epiphytes increased a little bit over the summer, but fertilization didn't have like a strong effect. But in our sites in Baia, there was actually a really strong effect of fertilization. And to show you this, you can see it here on the left panel, there are the plants from Ischia, and on the right panel, the plants from Baia. So we scraped off the epiphytes from the leaves, and you can see already there are much more in Baia, and also a lot of brown algae. And in September, for example, there was a red algae community developing on the plants from Ischia, and Pozzuoli was still only brown algae, which is an indicator for high nutrient availability. And then we measured also some morphological parameters. So here on the picture, you can see actually how big such a Posidonia oceanica shoot is. We looked at number of leaves per shoot, and there was like a strong decrease in Baia, and in Ischia, they remained roughly the same. We also looked at leaf height, which is the average amount of leaves from each shoot taken, and at maximum leaf canopy height, so always the largest leaf we could find for each shoot. And it was always leaves were always much longer in Ischia, and the difference was even more pronounced after the summer heat peak. We also looked at the leaf area index, which was almost at all times more than double as high in Ischia. And leaf area index is actually a really good indicator because it's a combination of the individual plant rates, so leaf width and leaf height with population level metrics that is per like shoot density. So it's a good predictor that tells us about changes in ecosystem primary production. And in terrestrial ecosystems, for example, they use also these indicators, maximum canopy height, leaf area index, and leaf coverage, because they were shown to have positive effects for ecosystem primary production. And if you know these indicators are impacted, this might have long-lasting and devastating effects on important ecosystem functions, such as the carbon sequestration capacity of the seagrasses.
Stephanie, 3 minutes.
Okay. Yeah, we also looked at the carbohydrates from both sides. Sugar in the rhizomes and starch in the rhizome, and we could see sugar concentrations decreased in both sides, and the decrease was more pronounced with fertilization. And starch contents actually increased in the low impacted side, but showed no difference in Baia. And with fertilization, there was even like a small decreasing trend. So this is actually really bad because there's no seasonality in the plants from Baia. And it is expected that rhizome starch concentration would rise because it's important for the winter survival of Posidonia and their growth in spring when there are still low light levels. So basically we found that the additional nutrient enrichment combined with the high summer temperatures had more deleterious impacts on seagrass plants from Bayer. And the less impacted sites were also affected by the high temperatures, but the effects of nutrient were not as clear. And our proposed indicators for future monitoring efforts would be percentage cover, leaf area index, maximum leaf canopy height, rhizome starch and sugar concentrations, and epiphyte biomass. So overall, basically, you saw we combined, we looked at the combined effects of temperature and nutrient stress on a small-bodied colonizing seagrass and a large-bodied foundation species. And you could see a Halophila as the more tolerant species that shows high plasticity and is able to recover faster. And Posidonia, on the opposite, has very limited colonizing abilities and slow growth rates and low reproductive capacity. Like basically what these results mean for the whole ecosystem, this will be shown in the next presentation by Pedro and Hauke that included such parameters in a model. And basically I want to thank all the partners. It was like a huge effort, this field work. So everyone that helped from the Stazione Zoologica, DETC, and Navarra Science Center, IUI, and also, of course, from the Leibniz Center for Tropical Marine Research. And of course, all the divers that were so amazing and made this field work possible. Yeah.
Okay, Stephanie, thanks a lot. Very, very nice presentation, perfect in time, 18 minutes. We can go to the next presentation of the Scenarios project. That will be about modeling of Halophila stipulacea dispersal and interaction in the Mediterranean. And okay, are you going to present or will be Pedro?
I'll present.
Okay, you will present. So, okay, Reuter, and then please, we can open the field for questions after this second presentation. It's to you, Hauke.
Okay, thanks, Gabriele. Hello, everybody. I'm Hauke Reuter from the Leibniz Center for Tropical Marine Research, and I'm going to present today on our modeling work. Actually, it's a work of Pedro Carretero, but he cannot present today, so it's my duty to do this. So we are going to talk about the biogeographical patterns and biological interaction of native and invasive seagrass species under the current and also predicted climate scenarios. And this is part of the Scenarios project in collaboration with Israel and Italian partners, as Steffi already said before and Gabriel noticed. So with our part of the projects, our research aim were to, there were quite a number of aims we had, and the first one was to represent and analyze seagrass reactions to changing environmental conditions by using ecological models. And this required that we integrate knowledge from the project, fieldwork and lab work, as well as from the literature. There's quite a lot of additional knowledge known, and this is also relevant for the models we are developing. And then to apply these models to analyze how species could respond, how they interact on the long term, and what effects climate change has on the community composition. For this, we developed two distinct approaches. One is the species distribution model, and the other one is the cellular automaton model, and I will explain both in the following, starting with the species distribution model. So for the species distribution model, we were interested to project the habitat expansion for tropical seagrass species in the Mediterranean. It's well known that this will happen, is already ongoing, and it's relevant to know how this will progress with climate change. And for this part of the project, there were actually three different study topics or questions we had to answer. One was to improve our understanding of the trade distance and relatedness between Halophila stipulacea and Halophila decipiens population from the Mediterranean Sea and from other locations worldwide, to see how they are related, what are the different traits, and to compare the origin of the Mediterranean species. And then the second important part was to see how the increase in sea surface temperature over the last 100 years can explain the colonization by Halophila stipulacea. So this is actually a hindcast to analyze what has been happening in the last century, and then to do a forecast and see how the habitat changes for both species during the coming century. So how climate change progresses and how this changes the habitat for the two investigated species. And with this, we also used a spatial distribution model. And both of this was relevant as input conditions for the cellular automaton model. So this was a precondition to develop the CA model. So the first of the questions was trade distances. So here we used a principal component analysis, a PCA, and a cluster analysis, and compared minimum, mean, and maximum temperature, as well as min and max salinity for different locations worldwide, where we find either Halophila stipulacea or Halophila decipiens, and to see how they cluster and how they are related. That's one, and this is actually the input and gives information on the species response to temperature or to salinity, which is essential for the definition of a potential suitable habitat in the spatial distribution model. For the hindcast model, we could only use sea surface temperature because the data on salinity was not available on the broad scale for the whole Mediterranean. And if you look at the figure, you can see the hindcast for 1920, 1950, 1970 represented with different colors, and the purple dots show locations of Halophila stipulacea, which you can find present. And you can see that many of the locations are represented quite correctly with a simple hindcast model, only using sea surface temperature, but some, mostly around Greece, are not presented correctly because the spatial distribution model only relates to one factor, temperature, and salinity is missing. Other environmental factor, biological interactions are not present in this case. But nevertheless, it shows the extension for quite some parts quite well. For the forecast model, we used sea surface temperature and salinity. And this looks quite differently. Again, for Halophila stipulacea, the purple dots show the present location, and you can see that implementing the model for the present conditions, shown in green, shows quite well coincident of habitat, suitable habitat, and availability of this species or presence of the species. And for the further years, you can see an extent to the west and to the north. And we did this actually for two different climate change scenarios, IPCC scenarios scenario 2.6, and which is more moderate temperature increase, and the 8.5 scenario, which is a very drastic temperature increase. And for both, you can see the habitat extension for Halophila, stipulacea, so it might move westwards and eastwards. It's not said that it will do this, but there's a possibility that it can move. For Halophila decipiens, the situation is a bit different. Currently, there's only one location in Greece where there's a population of this species. However, looking at the present habitat suitability, the species could also occur in the eastern part of the Mediterranean, but with increasing temperature under both scenarios, the potential or the habitat suitability increases quite a lot for actually most of the Mediterranean coasts. So for main conclusions for the spatial distribution model, so the principal component analysis suggests a similarity of Halophila stipulacea populations of the Mediterranean and the Red Sea. This is in accordance with the literature. What is suspected that Halophila stipulacea invaded the Mediterranean from the Red Sea for the last century. Nevertheless, for Halophila decipiens, the Mediterranean population is more related to the Atlantic populations, actually. The habitat suitability for Halophila stipulacea will keep expanding westwards and northwards in the Mediterranean as this part becomes saltier and warmer. And for Halophila decipiens, as you saw in the graphic, at the moment there's a restricted habitat suitability in the southeastern part, but with changing environmental conditions, there will be a very progressive and fast expansion of the habitat, potential habitat to the west and to the north, so that it will cover about 85% of the Mediterranean coast. So it will be very interesting to see how this species will develop in the next year, actually in the, whoops, that was too fast, in the next years. So we see that there's a high potential for a rapid expansion of non-native seagrasses in the Mediterranean, and there's a high potential that this will alter the Mediterranean seagrass communities. And this could also have or will certainly have a massive impact on the ecosystem functions and services. And to study this change in communities in more detail, we developed a cellular automaton model, where we start actually with the hypothesis that the Mediterranean seagrass communities will experience a transitions. And this transition will be from more long-lived and large species, Posidonia, as you saw in the previous presentations, and the communities will change to more small and fast-growing species as climate change progresses. So our objectives or aims were to investigate the synergistic impacts of climate change, so change in environmental condition, and also the presence of the invasive species Halophila stipulacea in the Mediterranean, and to see what effects this has on the community composition. And we used a cellular automaton approach for this, because this allows to include spatial interactions and the interactions between the different species. In our case, this is Posidonia oceanica, Cimodocea nodosa, and Halophila stipulacea as the invasive species. The other two ones are the present species. Yes, and how this coincides with the impacts of climate change. So I'm not quite sure if you're familiar with the concept of cellular automaton. So I will explain very briefly. So cellular automaton has actually, as the name says, cells which are combined to a grid as a very basic component. So you can have spatial interactions, and this can be in 1, 2, 3, or n dimensions. Normal or easiest to handle is a 2-dimension. Each cell will have a specific, or can have defined states. In the easiest case, or very simple case, this could be dead or alive or anything else. In our case, this will be different species. For instance, a cell has a neighborhood, which is considered when the rules are applied. And the rules actually are the most complicated things in the cellular automaton. So you have to define what happens in every situation, combination of neighborhood and state of the cell, how that changes. And this generates the model dynamics. And then you iterate. So you iterate or update the model over times, and you get different generations or time steps. If we shortly move to the left, there's a very old cellular automaton already developed in the early 70s called Game of Life. And here it's quite easy to see different rules. Rules are very simple. So if you look at the focal cell on the left-hand side, which is has a red boundary, so we are only considering the focus cell, and there are different rules how this focal cell changes. For instance, if there are two cell states, green is alive, white is empty. So if there's a green cell and there are less than 2 green cells or live cells in the neighborhood, then the cell will die. If there are 2 or 3 cells in the neighborhood, it will stay. If there are more than 3, it will die. And only in the case if there are exactly 3 cells next to an empty cell or a dead cell, then this cell will turn alive. So you see there can be very, very simple rules. However, even these simple rules will create very complex dynamics. There are a lot of applications actually to cellular automaton which are not feasible or very difficult to approach with standard modeling techniques. Mostly in the case where spatial neighborhoods are important. So you get very complex dynamics, often from quite simple to implement rules. And that's what we used for our Seagrass model. Actually interesting, we had suspected that there would be quite a number of spatial models for Seagrass, but we didn't find any. So this is something which is quite new actually, to implement a CR model for Seagrass. And we are quite happy that we took this approach. The rules which we implemented are for one, the first one is a response to the environment. So how the considered seagrass species react to temperature or salinity, how they grow, how they expand to neighboring cells, how species interact. So if there's one cell, for instance, with Posidonia, the other one with Halophila, and what will happen in this case if they'll just stop growing, or if one outcompetes the other one, and also some random events, plant fragments coming into the simulation, storms, sand waves, anchoring, heat waves, things like that. Oops, I'm too fast. And After implementing the cellular automaton, we developed different scenarios to simulate and see the outcome. So we used actually four areas across the Mediterranean as a basis. We had these 3 species, which I said. We have different environmental variables from min-max temperature, min-max salinity, and also heat waves, +1 or up to 1.5 degrees. We use the same scenarios as in the STM, so 2.6 and 8.5. And one cell is 0.2 square meters, and the whole area we usually cover is 40,000 square cells, and we use hexagons because of the neighborhood.
Okay, you have a couple of more minutes.
Yep, I need a couple of more, but not more. Thanks. So, and we have different maps for the initial seagrass spatial organization. So we used information, parameters from the spatial model, the STM model, from the field experiments, lab experiments, literature. So this from all the partners, literature which was available to see how these 3 species respond to the environment. What are the growth rates? This was mostly from lab experiments or literature, and also biological interactions between the different species. So what happens if they're very near to each other, occupy neighboring cells, so if one is out competing or the other, one will die, and information on this. So just some exemplary results. Actually, we are still evaluating all the results. There are a lot of simulations to be done. That's several thousands of simulations still partly to be conducted still. And this is one of the, you see here is one of the initial map we used for many of the simulations with 3 different species. You can see the cell state, possible cell states, dead mat of Posidonia, which is important, and the empty area. And this is one of the exemplary results for the moderate scenario for 3 regions, all of them with the same initial conditions. And you can see that depending on where you are in the Mediterranean, Halophila stipulacea will actually outcompete most of the other species, not completely in some cases, but Posidonia, under the given environmental conditions in the scenarios, will mostly, in most cases, will die back quite a lot. And you can see the temporal development over time in these graphs. So to come to the main conclusions from the cellular automaton, so with the model we implemented, we can represent the complex community dynamics in seagrasses. We didn't show so much about model validation, but we checked that the model works correctly. There are some situations where the interactions of seagrass have been investigated, and this is very well represented by the model. So for the future projection, it's obvious that there will be a replacement of the native species Posidonia oceanica by invasive and fast-growing species for most or many parts of the Mediterranean. Heat waves are quite important, and that triggered the collapse of the Mediterranean species in some cases, and then it was difficult for Posidonia to regrow in many parts. But what is also important is that the that the native Mediterranean species facilitate the dispersal of invasive tropical species. This is mainly through creating potential optimal environmental conditions, sediments, microbiota, where Halephora can settle and grow. And last not least, also important that the most western and the colder areas of the Mediterranean Sea could represent a refugee area for endemic Posidemia species. Okay, thanks a lot for listening, and thanks a lot to all partners for helping with all the information and providing information for us. Okay, thank you.
Thank you. Okay, I think this was perfect presentation to complete the exposition of the results of our two projects, because this is a perfect synthesis of what we are achieving from the experimental point of view, and then modeling what happens if our climate conditions will continue to be like that, and Mediterranean more opportunistic and faster species, more tolerant to higher temperature, can overcome the existing ecosystems with the clear damage for the ecosystem services that are provided by culinary species such as Posidonia. I think it is very, very, very interesting results. Okay, I open the field to questions, and questions can arrive from people connected on the YouTube channel, will be filtered to ask, or directly on the Teams platform in which speakers are connected. I have a question that has already been asked to Alex. Is on the, I think, from people connected in YouTube. And Alex, the question is, how long did the research take, and what are the applications? Maybe you can start answering to this question, and we wait for more further questions.
Yeah, okay. First, thank you for the question. And well, actually, the main experiment only lasted 2 months, but before that, there has been a lot of careful planning to set the conditions to be used in the main experiment. We have performed some trial experiments, so small, short experiments, in order to understand how the plant will, the conditions of the plant evolving in the mesocosm in the aquarium. And also we have collected a big amount of material, of biological material from roots and from leaves that we have a full time in the analysis of, after the experiment, in the analysis of the data. Well, sadly, there's been the COVID pandemic that has prevented us to come to the lab, but that has been a sad part of the project. But the application is actually to provide a set of tools to the management or the conservation programs of the species, and using the best technology available at the moment. That will be the main application of this project.
Thank you, Alex. Okay, Valerio.
I have several questions, actually. Thank you for the good presentation, very interesting projects. I start from the last. I say modeling of the findings in these studies appear to be useful to predict the future modifications of seagrass population into 2 sides of Mediterranean. But besides the very negative prediction for Posidonia, that I hope Posidonia will demonstrate to be wrong, I hope so. But how do they help in the correct management of seagrasses into countries like Italy and Italy? I mean, do you think that besides blocking the head, this can there is some other local activity that may change the disaster? And this is the first. Okay, I leave an answer for this, then I have some others if you want.
Okay, yeah, that's a very important question. Actually, what is still missing in the model is the impact of nutrients. This was something we wanted to implement, but we did not get so far. We will do that in future. Also for other locations, there will be an application in a master thesis for East Africa for the model, and in this term we will also implement nutrients. And then we will have to see how at least in the model, the species react to nutrients, and nutrients is something which is more local. Global change is not local, obviously. So management implication for global change are very general and nothing. No, there's a lot you can do local, but it takes very long. So that's something we will still have to evaluate. And I think what I presented are the very general results in a very short time. So if you look locally, the situation will be different, or could be different.
Oh,
that's something we will have to see. But at least it's a tool which you can apply locally with the given situation, with the temperature, and then sometime also with the nutrients impact, and then you can see how this will develop. Maybe there's no chance to change because these are natural or large scale problems, but maybe there's also something how to cope with it locally. But I can't answer yet.
And this brings to the second.
Valerio, sorry. Before you ask the second question, there was Guido raising his hand. Maybe you have another answer to the to the previous questions, or maybe you want to ask something.
I would just answer that we are trying to push the management and conservation authorities in Israel to set guidelines for water quality for seagrasses, because the nature reserve is concerned about a coral reef, which is slightly in a different location, and no one has really cared so much about seagrasses in the region. So if we are able to show that seagrasses are able to deal with thermal stress better if they're not polluted by nutrients, then there's a strong tool to say, okay, we can't fight climate change like Hauke said, but we can definitely contribute to the resilience potential by not killing them by nutrients, with nutrients.
Mm-hmm. Yeah, that's true. That's very interesting.
So besides the other merit of the program, it has the merit to bring some light on the seagrasses that are a little neglected from what I understand, moving some interest from the coral reef that are more well known by the large public. And it's important. But my second question came just from the answer, because especially in the first representations, these are for sure multi-stressor studies. From what I saw, apparently higher temperature coupled with the fertilization led to lower health, for example, of Arophila. I wonder if it will be possible to understand how this may be due to the internal physiology of the plant or to environmental changes, for example, bacterial growth or light limitation, as you showed by phytoplankton, that may influence the plant growth and expansion. And then I go even to the third that is connected, because nutrient can be one, but also currents, local currents may change the story. And in my opinion, in Baia, where you showed a higher effect of fertilization on epiphytes, it could also be due to lower currents in respect to Ischia, where you have an opened and more open area with higher currents. I don't know. This is two different questions. And I stop.
Who wants to answer first?
I don't know. You are all a group. So
I'll answer the first one, maybe.
You can start.
As we spoke before, and Hauke showed very nicely in the models, Lophila is going to be moving faster to the Mediterranean Sea, west part of the Mediterranean Sea. It already is in the Mediterranean Sea, and it's already in the Caribbean. And in the Caribbean, it's very aggressive, and it's taking over the local species. And in the Mediterranean, that hasn't happened yet. But if the Mediterranean is becoming more saltier and warmer and maybe more enriched with nutrients, then we are expecting and maybe already seeing a phase shift in front of our eyes right now already. So our title for the talk is called the Systems Biology Approach, and I did not show any bacterial work, but we have done a lot of work on bacteria as well. And we are seeing a change in the core microbiome of the seagrasses, and that means that the seagrasses are physically hosting different bacteria, and maybe they've lost the bacteria which are good for the seagrasses.
Mm-hmm.
So we think that the actual function of these bacteria which have gone might be important for the seagrasses for resilience. We know that microbiome help seagrasses in many ways. I won't go into that right now, but so I hope that answers some of your question.
Yeah, I see.
I have a similar answer because, I mean, we are approaching the study of the species response to the stressors we impose from so many points of views that our hope actually in the system biology approach, as Gideon was saying, is also using gene expression analysis or any other level that we are approaching, is to be able to disentangle what you were asking. So to understand if the response, the adaptation is due or any kind of results we observe is due to the species itself, to the change in physiology in the plant response, or is due to changes in the ecosystem in a wide sense. I mean, the associated communities, epiphyte cover, macrobiota, and so on. And regarding currents, yes, you're right. I mean, the current regime in our study in the Mediterranean can be an issue because Baia is surely less dynamic as environment with respect to Ischia. Though, I mean, differences are so evident and so strong that I don't think that's only current to have an effect, because epiphyte cover is incredibly is the amount of organic matter on the this rate of cost in Baia is much higher than in Ischia, and this is affecting the epiphytic cover on the leaves.
Okay. Okay.
Okay, if there are more questions, we can wait one more minute. Otherwise, we can close the session. We are almost in time with our schedule.
Gabriele, in the meantime, I inform that since somebody has, had an older version of the program, I inform everybody that we will start back at 3 with the group of Genoa. And now we will put also a fixed panel to the paper somewhere in our web place, so people can know that we start at 3. Not
okay.
3.
Yeah, it's 3 o'clock with the SmartX project, and then tomorrow morning at 10 o'clock with X-Sites.
Yes.
Okay.
But this was according to your last modifications of the program, and probably somebody did not.
As an old version of the program. Okay, so the new version is, we start at 3 o'clock.
3 o'clock. As we decided with the last version.
3 o'clock. Okay, so I thank everybody for being present following the session, and so we will meet again at 3 o'clock. And I wish you a nice lunch.
Yes, you are invited to the lunch session.
Lunch session is here. Person.
Peace.
Bye-bye. See you later.
Okay, see you at 3.
Thanks for all the speakers, by the way.
Thank you, Gabriele.